The next time we meet, we'll be discussing Chapter 11, and what I'd like to do is spend some time in the class going through some practice problems to give everybody practice with different types of monohybrid and dihybrid crosses for Mendelian inheritance patterns. So today, for your study guide, what I would like you to do is to watch this video, jot down any notes, and when we return to class, you have the opportunity to ask questions. We'll take a short quiz to cover your comprehension and make sure you've watched this video in its entirety, and then we'll jump into practicing problems in the class. Following that, then we'll return to the rest of the lecture material. So in the course of this lecture today, we'll be looking at Gregor Mendel and his work with the common garden pea to uncover some of the patterns of inheritance. We'll talk about some terminology like dominance versus recessive, and introduce you to both monohybrid and dihybrid crosses. This is primarily what we'll get more experience with in lecture time. We'll discuss what it means to be homozygous versus heterozygous, and what a genotype and phenotype are. Then we'll talk about the two laws that Mendel coined, the law of segregation and the law of independent assortment, and how they relate to the process of meiosis, which we already covered in Chapter 9. And I'd also briefly like to talk about how the laws relate to probability. What is the chance you'll inherit a particular phenotype given your parents' genotypes, for example. When we return in lecture, after we do some practice problems, then we'll go on to talk about Mendelian genetics as it relates to autosomal dominant and autosomal recessive traits and diseases. And then we'll extend that to other patterns of inheritance, like multiple allelic traits, incomplete dominance and incomplete penetrance, pleiotropy, polygenic inheritance, and then we'll also talk about sex-linked traits. Mendel used the garden pea as his model, scientific model for genetics. We'll talk about scientific models. You know, the common lab rat is used to discover information about human diseases, for example, and in this case, the garden pea was used to look at patterns of inheritance, which can be applied to many other systems, including human genetics. And so several of the reasons Mendel used the garden pea was that it was very prolific, produces many seeds with one cross, and so he had a lot of progeny to analyze. It's very hardy, so he could grow it in his garden. Didn't have as many problems with cold or reduced sunlight, so it's easy to take care of, and it was self-fertilizing, which means that he didn't have to manipulate it in any way just to self-fertilize, but there was the opportunity to cross-fertilize, so he could take specific male, specific female parents, cross them, and look at the progeny. And so you can see here, here's the petals, and then this is the female part, where pollen would land up here, and the sperm would travel down and fertilize the eggs in each one of these seeds, and then the seeds develop. This is the male part, and the pollen would come from there onto the female. So if you wanted to cross-fertilize them, you could take pollen from one male flower and then directly apply it to a female, and therefore ensure you knew what the parents were. Mendel looked at many different traits, including tall and short, which we'll talk about. He looked at pod shape of inflated versus constricted pods, round versus wrinkled seeds, the color of the seeds, yellow versus green, the location of the flower on there, and then color of the flower and color of the pea pod. And so he examined and kept track of a lot of different traits, and that was one of the great things about Mendel, why he's such a brilliant scientist, is he kept a good lab notebook, and he was very observant with his studies. And he kept track of, if he crossed certain individuals, like a tall plant by a short plant, what did the progeny look like? And what he came to find is that every time, every cross, no matter what trait he was looking at, he had similar results. Even though the numbers are quite different here, what they always resulted in was two different types, either tall or short plants, and you can see the ratios of all these end up being 3:1. This 3:1 ratio is the ratio between the dominant and the recessive trait. In this chart, what you can see here is the dominant traits are here on the left and the recessive traits are on the right. And what that means is what Mendel first did was to cross a dominant by a recessive, and they were true breeding, which means all the dominant traits, whenever they were self-fertilized, would only result in dominant progeny. The recessive, all the short plants, were also true breeding, so when you crossed a short plant by a short plant, you always got short plants in the progeny. So first he crossed these, and this is called the F1 generation. When you cross two parent generations together, you get the F1 generation, which stands for filial, the first filial generation. Then what he did was to cross two F1 plants, and so he ends up with F1 by F1, and that results in the second filial generation, or the F2 progeny. And that's what we're looking at here, are the F2 results. And so when he crossed that F1 by F1 to get the F2 generation, these F2 results always resulted in a 3-to-1 ratio of dominant traits, so the tall trait here, to the recessive trait, or the short trait. And we know it's dominant because most of the plants end up being in the 3 category. All of those are the dominant traits. And so the dominant to recessive was always 3-to-1. So here's an example of this cross. In the parent generation, we have tall plants, which we are going to, in the parent generation, we have the tall plants by the short plants. And so the tall plants here are illustrated with big T, big T. As I said, they're true breeding, and so they have 2 of the same letter to indicate T for tall. And then the short plants have 2 little t's to indicate short. Whenever we choose designation for these traits, big T and little t, you always use the letter for over the dominant trait. So the dominant trait here is tall, so we use a big T, but we don't use an S for short, we use a little t instead. And so this is his parent generation. And when we set this up, then we're going to cross these parent generations to get, like I said, the F1 generation, and then we'll cross 2 F1 plants, or siblings in this case, to get the F2 generation. So let's take a minute to talk about homozygous and heterozygous. We were working with plants that were big T by little t. Big T by little t, little t. What does that actually mean? Well, those designations reflect the genotype, or what the genes of an individual are. So if you remember that individuals, as we've talked about, most individuals are diploid, and so that means they have 2 copies of every chromosome in their nucleus, so that makes them 2N. And the 2 chromosomes here are illustrated with a red chromosome and a blue chromosome. A particular gene would be located at what's called a locus. So that is just the site on the chromosome where a particular gene is located. The alleles then are the versions of the genes that are located at a particular locus. So here in this example we have big G and little g. That could mean green pods, which is dominant, and yellow pods, which is recessive. So a big G for green and a little g for yellow. So an individual is homozygous, or true breeding, if he has 2 copies of the same allele. So 2 big T's, or 2 big G's, or 2 little g's, or 2 little t's, the same version. So that's homozygous. If an individual has 1 copy of each of the 2 alleles, so he has 2 different alleles at a particular locus, 1 on each chromosome, so big G, or as we saw before, big T, little t, those would be heterozygous. Heterozygous and homozygous then refers to what the genotype of an individual is. So what are the alleles that an individual has? What are the genes? The phenotype then refers to how the individual appears. So homozygous can either be homozygous tall or short, depending on if it's big T, or little t, and the phenotype for an heterozygous individual will always appear as the dominant phenotype, tall. What a Punnett squares allows us to do then is to create a prediction of what the progeny in the next generation would look like. So in this case, this heterozygous individual, when it forms gametes that are going to be, again, haploid, those gametes can either have, since they're only going to have one copy of each gene, they can either have a big T or a little t. And so what we do in a Punnett square is across the top, this is the female gametes, and the female, if a big T, little t heterozygous individual is the female, it can either have eggs that are going to be big T or little t. Same thing, since we're crossing by siblings, one parent is going to be the male and one's going to be the female. And so the sperm, the gametes for the male, can either be big T or little t, so we can separate them in the Punnett square. If by chance one big T and one little t, or one big T and one big T meet during fertilization, the resulting progeny will be big T, which means that that progeny is homozygous with the dominant trait, for the dominant trait, and will be tall. If big T and little t meet in either of these two instances, then the progeny are heterozygous for the genotype, but because they have a big T, and big T is for tall, and it's dominant over short, then those individuals will be tall also. If a little t sperm meets a little t egg, then we end up with a homozygous recessive individual where the phenotype will be short. And so this is where we get the 3-to-1 ratio that Mendel was seeing. We have 3 tall plants and 1 short plant. So these have a phenotype of 3 and a phenotype of 1 short. And so he was always looking at the phenotype. He didn't know anything about genes and alleles and chromosomes yet. He just called them factors. So what Mendel was able to deduce from all these crosses, the fact that he always saw tall plants in the F1 generation and he always saw this 3-to-1 ratio in the F2 generation, was that each individual has 2 factors for each trait. We know this is true now because the laws of chromosomes and meiosis, we know that each individual has 2 chromosomes, and on those chromosomes are those 2 genes, what Mendel called factors. And Mendel helped to formulate the idea that these factors segregate during the formation of gametes, so that we start with an individual that has 2 factors, and then the gametes would only have 1 of the factors. Each gamete contains only 1 of the factors from each pair, and fertilization then, when egg and sperm meet, gives rise to an individual with 2 factors for each trait. Going back to the idea of then the progeny would all be diploid. So this is Mendel's law of segregation, that the 2 alleles, the 2 versions of each gene, segregate during meiosis so that the gametes that result from meiosis have only 1 factor. With these traits, Mendel took it to another step. Not only did he look at single traits that were being crossed, but he also, so in the monohybrid crosses, but he also did dihybrid crosses, and that is looking at 2 traits in an individual being crossed. So here, the example that's given in the textbook is from an individual still dealing with the tall, so an individual that's tall and an individual that's short, and the second trait that we're dealing with is the example I gave before with a big G, little g, pods that are green and pods that are yellow. Again, these individuals are true breeding, which means if you self-fertilized any one of the individuals, all the progeny would be the same phenotype. So big T, big G, when you self-fertilize it, always gives the result of tall plants with green pods. The same is true for the recessive traits, little t, little t, little g, if you self-fertilize, they always give short plants with yellow pods. So the parent generation is shown here. These in the gametes, during the formation of gametes, these alleles will segregate, as we just saw with the law of segregation, so that each gamete will only contain 1 of each of the 2 factors. So the gametes here contain 1 T, big T in this case, 1 T, little t in this case, and then also 1 big G and 1 little g. These 2 gametes, when they meet in the F1 generation, give rise to an individual that goes back to being diploid and now is heterozygous at both loci, heterozygous for the two alleles for the tall and short, heterozygous for the two alleles for green and yellow. And because tall is dominant over short and green is dominant over yellow, this individual that is a double heterozygote ends up with a phenotype of tall with green pods. Before we do the dihybrid cross to cross the two F1s, in other words, we'll be crossing an individual that's big T, little t, big G, little g, by big T, little t, big G, little g. And then we're going to look at the resulting progeny in the F2 generation. But before we do that, I want to take a look at Mendel's second law, which is the law of independent assortment. Here with the law of independent assortment, Mendel stated that each pair of factors segregates independently of the other pairs. In other words, the factors for tall will segregate independently from the factors for green. And so all possible combinations of factors can occur in the gametes. The way that's realized when we do a Punnett square, again, this is the Punnett square for a dihybrid cross, we end up with all possible combinations of gametes. So the only possible combination that can form from this individual is a gamete with a big T and a big G. And the only possible combination that can form from this individual is a little t, little g. But when we're looking at the the F1 progeny, there's many different gametes that can form. And all combinations are possible. So we can end up with a big T with a big G. We can end up with a big T, little t with a big G, big T with a little g, and little t with little g. In other words, T segregates independently from G, and so we can end up with all four possible combinations in the gametes, where every gamete has one T and one G. We line those up for both the female eggs, or gametes for the female, and the male gametes, the sperm, and then we fill in our Punnett square where we bring big T, big G down and big T, big G over, and we end up with an individual that is big T, big T, big G, big G. The genotype for the individual are the allele designations. The phenotype would be tall, because it's homozygous dominant for tall, homozygous dominant for green. So the phenotype is tall with green pods. We can further fill out the whole Punnett square by bringing the gametes over and down, and you end up with all the possible combinations in this 16-square Punnett square. In this example, the squares are color-coded, and you can see that the light yellow color indicates an individual that's tall with green pods. The gray colors shows individuals that are tall with yellow pods. The purple shading shows individuals that are short with green pods, and in the final example that's green shaded, you end up with individuals that are short with yellow pods. This is an example of a double homozygous recessive. If you add up the phenotypic ratios, what you end up seeing is that the ratio will always be 9, the yellow shading, to 3, the gray shading, to 3, the purple shading, to 1, the green shading. This is a classic example of a dihybrid cross where both traits are Mendelian traits, and you always end up with a 9:3 to 3:1 ratio in the F2 generation. Okay, we'll get some more practice with monohybrid and dihybrid crosses, but for a moment I want to show you how these two laws of Mendel's, the law of segregation and the law of independent assortment, are ultimately realized in the process of meiosis, which you've already talked about. This figure from the text shows the steps of meiosis. What we see is after the chromosomes duplicate and the chromosomes line up at the metaphase plate during metaphase 1, you can see they're duplicated, and remember the red indicates coming from the male parent and the blue indicates the female parent, and then you can see the size of the chromosomes indicates they're two different chromosomes. So top we would have chromosome 1, and then down here we'd have chromosome 2. They line up at metaphase plate, and what the law of independent assortment says is that the chromosomes can orient in one of two ways, in this case with two chromosomes. If you have more chromosomes, there will be more combinations of how they can line up. And so you can see here that for the small chromosome, you can have the red on the left and the blue on the right. or for the large chromosome, the blue on the left and the red on the right. Or they can line up in the opposite orientation where both red chromosomes are lined up on the left and both blue are on the right. When this splits during anaphase and telophase, then each gamete ends up with one duplicated copy of each chromosome. And you can see that there's 4 possible results. You can end up with individuals that have one of each, or you can end up with individual gametes that have both red and both blue. Then these factors segregate from Mendel's second law of segregation, and you end up with 8 possible gametes, 2 of each kind, because they're exactly duplicated copies, where you have 1 red, 1 blue for the dominant traits here, 1 red, 1 blue for the recessive traits, or you have 2 reds and 2 blues. And so you end up with 4 possible combinations because of the law of independent assortment and the law of segregation. So the law of segregation is realized essentially, you could say, in anaphase or telophase 2 of meiosis, the second stage of meiosis. And the law of independent assortment is realized during metaphase 1, when those homologous chromosomes line up at the metaphase plate. Now that we've talked about Mendel's 2 laws, the law of segregation and the law of independent assortment, we can briefly take a look at probability. We won't get a chance to apply probability too much, but when we get to more complicated genetic problems, perhaps in upper level courses, it'll become very handy. So I just want to mention it by way of introduction. So here we have a cross for the trait that refers to unattached or attached earlobes. And you can check your ears right now. Unattached earlobes are the dominant trait, and attached earlobes, where the lobe connects to the ear, the lobe of the ear connects to the ear jaw, that would be attached earlobes. And so the parents of this cross are both heterozygous, which means they have 1 big E allele and 1 little e allele, and the phenotype then would be the dominant trait, and that is unattached earlobes. When we look at the cross in a monohybrid cross, we would have each parent contributing 1 big E and 1 little e in the eggs and the sperm, and so you can see the resulting progeny would have 1/4 chance of being big E, 1/4 chance big E, 1/4 chance big E, little e here, and then 1/4 chance of being little e. So 3 individuals will have unattached earlobes to 1 individual with attached earlobes. We can apply a couple of probability rules. The first one is the product rule, and this is really looking at genotype. And what we can say is that because an individual in the progeny has 1/2 chance of getting big E from father and 1/2 chance of getting big E from mother, they have a 1/4 chance then, if you multiply these together to get the product, of having big E, or being homozygous dominant. So that would be an example of the product rule. The sum rule then illustrates how you configure probabilities for phenotypes as opposed to genotypes. So let's take a look at that. So as we saw before, using the product rule, we calculated that there was a 1/4 chance of an individual having the genotype of big E. If we followed the product rule similarly for the other phenotype or genotypes, we would see that there's a 1/4 chance of an individual getting a big E from mother and a little e from father, a 1/4 chance of getting a little e from father and a big E from mother. So there's 2 different ways to look at it. Now we normally don't write little e then big E in that convention, but in this case I'm just illustrating that we have 2 possibilities of how an individual could end up being heterozygous. And so if we add these up, as we sum them in the sum rule, you end up with a 3/4 chance of an individual having at least 1 big E allele, and therefore being the dominant phenotype of unattached earlobes. So this is a pretty simple example of the product rule and sum rule. We can just as easily draw out a Punnett square and figure this out, but in more complicated genetics problems, especially when you're looking at 2, 3, 4, 5 different genes, trying to look at combinations of traits, then it's easier to apply the product rule and the sum rule. Okay, so next time we meet in class, we'll be applying this information. We'll take a short quiz and then we'll apply the information by doing some examples of genetic problems, and also we'll talk about sex-linked traits and how to set those up in Punnett squares and solve those problems.